Good morning, everyone. Welcome to the Clinical Research Ethics Seminar Series. We're very pleased today, are expecting a very strong turnout. So we have actually a presentation this morning, an invited speaker, and we'll be following that by a panel discussion on the same topic today on placebo. I'm Carol Fetter from the Center for Clinical Research, and we're very pleased today to have put this program together. It's presented by University Hospitals Case Medical Center and Case Western Reserve University. So thanks to all of you for supporting us in this effort. And the program has been approved for continuing research education credits. We know that's important to everyone as well. So there will be 6 CRECs awarded for the program today. Please be sure that you've registered it at the front so that we can make sure you get your credits. And also, we would just ask for today, we are going to be recording the session, so at the point that we would have open questions, we would like you to use the microphone, and also, if you could please turn off all of your portable devices. I won't name them all, but I'm sure you know what they are. During the presentation this morning, we would really appreciate that. So it's my pleasure today to introduce to you Dr. John March, and he is a professor of psychiatry and psychology, and also the director of the Division of Neurosciences Medicine at Duke Clinical Research Institute. Dr. March develops, conducts, and facilitates research projects in adult and child neurology and psychiatry, ranging from biomarker, biosignature development and validation, imaging, genomics, phase 1 through 4 industry, and federal clinical trials, and also including outcomes research. We're very pleased to have Dr. March here with us today. Please join me in welcoming him.
Can you all hear me? It's a pleasure to be here. I was surprised to get asked to give this lecture, although I have done a lot of clinical trials, many of them involving placebo, and actually chaired a research forum for the American Academy of Child and Adolescent Psychiatry that was published in 2004. Forum was in 2002. I don't claim to have any particular expertise in placebo per se, or in the ethical context in which placebo unfolds. So I did my best to talk the folks here at Case out of inviting me to give this lecture. And as you see, I was completely unsuccessful. I have no idea why that is. So I decided what I would do is talk with you a little bit about a clinical trialist view of placebo in randomized controlled trials in psychiatry. And although because I'm a psychiatrist and my research program has been largely in psychiatry and until recently, and now it spans all of neurosciences medicine, most of what I'm going to say is applicable to other therapeutic areas as well. And I hope you'll, those of you that are not primarily interested in psychiatric research will also find this interesting and provocative. So these are my disclosures. I work a lot with industry, although the lion's share of my funding is from the National Institutes of Mental Health. I have one company that I work with, Metavante, that does centralized ratings, where I'm on their scientific advisory board, where I hold equity in the company. All of these other industry contexts or opportunities to work together collaboratively in the public-private partnership space are strictly on the science side. So for 15 years, I have not been on a speakers bureau, done any speakers training, have actively worked to avoid and publicly promoted avoidance of engaging in marketing-driven activities with industry while still trying to preserve as best I could the opportunity to do science-side collaborations. So if you wanted to see all of this, it's actually up on the web for anyone to take a look at. So the first question I thought we ought to address is, what is a placebo? And having addressed that, we can move on to talk about regulatory context, a little bit about decomposing the placebo response. It's actually gotten very interesting. Thinking about how one might minimize the placebo response, And there are a variety of good reasons for doing this that are both bioethical as well as pragmatic. To talk about when a placebo is necessary, and then we'll finish up with thinking through some of the ways that we can either modify placebo-controlled trials or get out of the business of having to do placebo-controlled trials altogether. So this is a placebo. It is a pill. It will not have, in a randomized controlled trial, the word placebo stamped on it, because the purpose of this particular gadget is to provide an opportunity to mask or blind the nature of the treatment which the patient is receiving to both the doctor and the patient and all the other personnel that are involved in the clinical trial. So the old view of placebo is that is what it did. It was an inert sugar pill that looked exactly, tasted exactly like the active treatment, but otherwise was inert. The new view of placebo is that it is an active intervention that in many cases, at least in neuroscience, activates exactly the same circuitry that is activated by the active ingredient of the treatment. It just doesn't do it as well. And I'll show you some really intriguing imaging data that supports that view in a moment. As I said, it looks like the active treatment. This is what's called masking, and allows blinding of the patient, doctor, and rater. This is what we mean when we say double blind. And it's supposed to, by intention, control for all of the things that happen to the subject in a clinical trial that are related to participation in the trial, other than the active ingredient of the treatment that's being tested. So all this is old hat and probably familiar to all of you. So that's what a placebo is, but it's not the same thing as the placebo effect. So the placebo effect is defined as the response that's associated with placebo. So placebo subjects can get worse, they can stay the same, or they can get better. And typically when we think about the placebo response, it means improvement on placebo, but in fact the placebo response can involve harm's neutral response, nothing happens, or worsening. So it happens in a large proportion of the patients who are enrolled in clinical trials. It's not remotely trivial and needs to be thought through carefully in the sense of experimental design and the operational considerations that are involved in doing clinical trials. The effect can be positive or negative. The negative effect is typically called the nocebo response. It involves subjective endpoints. The patient, for example, might say, you know, I have less pain. And it also involves objective response. You might actually do a pain challenge and look at an objective marker, a biomarker of response. And the placebo response is common across all therapeutic areas, with some exceptions. So, for example, in the cancer world, placebo responses are not as worrisome or bothersome as they might be in psychiatry. And moreover, in some areas, like cancer is a really good example, you wouldn't ordinarily think that it would be ethical to do a placebo response, because withholding active treatment is, in patients who are at risk of death, is simply not something one would do. So there are areas where you might think about using placebo in a therapeutic area like cancer, but for the most part, it's used in areas like in neuroscience, where you really have a possibility of benefiting from the placebo, and that response needs to be disentangled from the treatment response in the active treatment group, for which it serves as the comparator condition. So placebo is not the same thing as no treatment. There was a meta-analysis done recently from a group in Finland, a Cochrane review, that compared 52 studies that included both placebo, an active comparator, and a no treatment control condition. And if you look at the placebo condition relative to the no treatment control condition, there was relatively, in this group of studies, relatively little difference. So they concluded that there was no evidence that placebo interventions in general have clinically important effects. That may not be quite as true in the neuroscience world, where the placebo response is more robust. If you actually ask practicing clinicians, have you ever prescribed a placebo, about 6 in 10 will say yes. So this would be to give a patient a medication, something innocuous like a little bit of Benadryl, because the value of doing that, the expectancy associated with the placebo response, is not inconsiderable. In fact, when you watch physicians in psychiatry manipulate medications, why don't you raise this one a little bit and lower that one a little bit and call me in a week, what they're doing is capitalizing on this effect, this expectancy effect, and also on sort of regression to the mean. Somebody who's worse is likely just with the passage of time to get better, both components of the placebo response. But in general, there's no reason in medicine, based on the data, to say that we should have formal placebos that we can give to patients as alternatives to active care interventions. Now, what about the regulatory context within which we use placebo? So you all know that the Declaration of Helsinki, which was most recently modified in 2002, and the usual rule, this is true across all the different bodies that have proposed standards for doing placebo-controlled trials, the usual rule is that you should compare the new treatment against the best available treatment. That is, if you can avoid placebo because you have a standard of care and you have a new treatment, the idea would be to see if the new treatment is either equivalent in a test of equivalence trial or non-inferiority trial, or beats the currently best available treatment. And it's the latter model that's typically used. For example, in cancer, the old treatment is now run either against or on top of the current standard of care. So placebo is only permissible when there's no other alternative. If you have a standard of care, as I'll talk about in a moment, in some sense it actually makes more sense to do an active comparator trial. There are exceptions that the Declaration of Helsinki endorses, which are common in other regulatory contexts as well, and that is when the condition is minor and the risk is minor, and there's a compelling scientific rationale, then using pill placebo in a clinical trial is a reasonable thing to do if it's required in order to make that experiment scientifically credible, which of course is part of the ethical context in which clinical trials unfold. The International Conference on Harmonization standard, which is the one that the FDA tends to like better than Helsinki because it's a bit more user-friendly for placebo-controlled trials and is more widely applied in a North American clinical trial context, where the Europeans tend to look a little bit more toward the Helsinki Declaration. Says that placebo is acceptable when there's negligible risk for harm to subjects. So if you had an expectation in the nocebo response that the placebo was going to produce a serious adverse event, then it would not be ethical to use placebo. So the placebo can't harm the patients directly, or if withholding active treatment would harm the patients. So you'd have patients who are at risk of dying in the most egregious example, then it would not be ethical to use a placebo in a clinical trial. It's also reasonable to use a placebo when you have really nasty side effects with the active comparator. So now you're in a placebo-controlled trial increasing assay sensitivity, which I'll define for you in a moment, so you can expose fewer patients to the active treatment by using a placebo control condition, and this makes placebo actually warranted. Or if you have an active comparator that you would like to use, but it's actually not credible as a comparator treatment. So one of the options that I'll talk about to placebo is using a dose-controlled comparator. So a little bit of drug, and then a little bit more drug, and then a little bit more drug. But if that lower dose is not really credible as an experimental condition, or if it's potentially associated with other forms of harms, then it would not be, it wouldn't be credible as well, and so a placebo condition would be warranted. Now, placebo is unacceptable in this regulatory context. If you put it in the negative, when there's no chance of benefit from placebo, when you expect adverse events associated with placebo and they're clinically significant, when the disease being treated is expected to worsen over the course of the clinical trial, and that worsening would expose patients to risk well above the minor harm standard, and when there's a standard of care that's available. This is the Helsinki definition, and the way it's typically handled in this country is to say that, in fact, we don't have standard of cares, at least in most neuroscience contexts. Now, in the epilepsy world, for example, it's like the cancer world, so that patients would have seizures if you didn't treat them with an active drug. There are active comparators that are available. You can run the current generation treatment against the previous generation treatment, the next greatest thing on the block to the current treatments, which are clearly unsatisfactory. Otherwise, there'd be no reason to do a trial in the first place. So there are contexts in neuroscience that fit this standard of care definition. The only drugs in psychiatry for which we could make an argument for standard of care are the psychostimulants in ADHD, where it's no longer possible to do a meta-analysis of clinical trials because there's so many of them. And in fact, now across the age range, there are over 500 reviews or meta-analyses of clinical trials, so much so that Jim Swanson a few years ago wrote a review of reviews. He did, in essence, a meta-analysis of meta-analyses. And when you get to that point, you begin to think that maybe there's a standard of care at play in the field. But even now, we do placebo-controlled trials in ADHD because it actually improves the scientific credibility of the experiment, and because the trials are short and there are rescue procedures, things we'll talk about in a bit, it's ethically reasonable to hold subjects on placebo within a clinical trial for ADHD, and by doing so, you actually learn some things that are very important that could be learned in no other way. Now, placebo in an adult trial context has important implications for informed consent. So if you have a person who is decisionally challenged for whatever the reason, whether it's low IQ, language difficulties, whether the disease being treated impairs their ability to make decisions, for example, say Alzheimer's disease, whether a level of consciousness that's associated with the illness, a patient who is psychotic and delirious, for example. All of these things would make the part of informed consent that's required to explain to the patient that they would possibly receive a placebo and what that would mean has to be factored into the decision of whether a placebo is acceptable in the clinical trial context. Here, the critical thing is to avoid the therapeutic misconception. And this happens in a research context where patients and not coincidentally, persons who work on the clinical trial, faculty and the operations teams, all believe that they're helping the person who is in the clinical trial. That is, the expectancy is that by being in the trial, it will be of advantage to you clinically. Now, there is some truth to that. Being in a clinical trial, you get better assessments often. And in a placebo-controlled trial, there's usually the expectation that you could improve even if you were given placebo, although the directional hypothesis would say that you wouldn't improve as much as if you were on the active treatment. That's the experimental question. But even so, clinical research is not the same thing as clinical care, and one needs to explain to patients, and I think also to hammer home to those of us who do the clinical trials, that clinical research is not the same thing as clinical care, and we actually have to directly confront this fallacy of therapeutic misconception as part of the informed consent process. That is a subtle distinction, and if you actually look at subjects who've been consented, and there's a lot of research about this, most subjects actually don't get it. So we're lacking something in the way that we do this informed consent process if we want to make sure that as part of the process, patients understand that they are participating in research and there isn't an expectation that they will necessarily benefit from it. On the other hand, there is significant benefit to society for participating in research. And so in our clinical setting, when I ask patients, would you like to be in a research project? And I ask them, why did you decide to participate? They will sometimes say, well, I can get something here that I can't get anywhere else. So if you have a novel treatment or you're the only player in town that can provide a particular form of treatment, that's an incentive to participate. You get better assessments and much better monitoring than you would typically get in a clinical context. That's a reason to participate. But what they tell me most of the time is they want to take something about their illness, something about the suffering that they incur as part of being mentally ill, and to transform it into benefit for other people who also suffer. And so there's a significant benefit to being in a research project that involves learning something useful. And that needs to be explained to patients as part of this informed consent process. That is, you may not benefit. That's the therapeutic misconception. But there will be, by participating in research, benefit that goes beyond you. And all of these things require a certain level of mental acuity that has to be factored into the decision about whether a placebo-controlled trial is in fact ethical. Now, this is compounded in the pediatric world because you not only have to get informed consent from the parent or guardian, you have to get assent from the subject. And the informed consent process is more complicated in this context because kids don't have the same neurocognitive decision-making capacity, just in a developmental context, than adults do. Now, you could argue that an adult with an IQ of 70 is going to be more impaired in this sense than a teenager with an IQ of 140, and that may well be true. So chronological age doesn't necessarily have anything to do with neurocognitive computational age, the ability to understand. But the basic principle is that you have to think through the consent process with respect to not only the subject of the trial, but the parent or guardian as well. And so we have two consent forms with different language, and it just gets very complicated. Now, the federal regulations actually have language in them talking about this, when you can do pediatric trials that include a placebo arm. This is part of the FDA enabling language. And they require minimal risk. Sort of minor, more than minor is a tough sell in a pediatric context. You can have minor increases over minimal risk, but only if there's the possibility of direct benefit. And we'll talk in a moment about what that might look like. And you can have minor increases over minimal risk and no prospect of direct benefit. This is the toughest of the standards. When the trial itself is so important because it's going to yield generalizable knowledge that if it can be done in no other way, if that generalizable knowledge, which has significant societal benefit, is present, that decision context is present, then a placebo could be considered ethical, even when there's more than minimal risk and there's no potential for direct benefit to the research subject. Now, this is kind of a way to think about prospect of benefit, since this comes up all the time when we think about whether it's okay to use placebo. One is that benefit essentially is evaluated relative to what's called clinical equipoise. So clinical equipoise means at the group level that I have no idea which of these treatments is going to be better. I may have a directional hypothesis that says the active treatment is going to beat the control treatment at a 20% test of differences. 60% of patients will respond in the active drug and 40% will respond in placebo, and so I've powered my trial on that test of differences and have 100+ subjects in each arm and so on. So there's a clear sense that the trial is organized around the expectation that the active drug will beat placebo, the directional hypothesis. But in fact, I don't know the answer to this question. Otherwise, I wouldn't be doing the experiment in the first place. And at the group level, that's called clinical equipoise. It translates to the subject level with something called the uncertainty principle. And that is when I sit with the patient that I don't know with that particular individual whether they're going to do better, they're going to stay the same, or they're going to do worse with respect to which arm of the trial they're assigned to in the process of randomization. So if you have a situation in which you don't know the answer to the experimental question, and you can't say to the subject, you know, I don't know what's going to be of these options, which one will be best for you, it's actually pretty easy to sell a clinical trial because the investigators are motivated to learn the answer to the experimental question. The patients and parents are motivated to learn the answer to the experimental question because it will benefit them going forward and also other people like them. And it provides a kind of a context within which you can explain this issue of therapeutic misconception and also talk about the possibility of direct benefit. Now, it should be obvious from that discussion that if you have a standard of care, that you're not at clinical equipoise and there's no uncertainty at the patient level about which would be better. So if I have a treatment which is, as is true, for example, in the cancer world, which I know will benefit patients. They will die if they don't get this kind of treatment. I don't have any idea whether an alternative treatment is better than the accepted treatment, even when the whole trial is running against the background of conventional treatment, say adjuvant therapy for breast cancer. It's pretty hard, given that context, when you have a standard of care. Everybody knows that this thing has to be given to patients because there's unacceptable harms if you don't. It's pretty hard to run a placebo-controlled trial in that context precisely because you're not at clinical equipoise and you can't communicate uncertainty about what works and what doesn't work to the patient who's being consented. So a direct benefit can accrue even in the absence of a standard of care, which is most of the time what we deal with, in several different ways. One way is the expectation of improvement with placebo. The second way is you actually benefit usually from placebo, even though you can have nocebo effects. You are less likely to have adverse events with placebo than you are with the active drug because it's the active ingredient in the drug which is causing the adverse events in the first place. There's benefit from assessment and monitoring during the course of clinical trials. And as we talked about a moment ago, there's benefits from participating in clinical research, including delayed benefits that will not show up necessarily during the period of the clinical trial. And here's an example of a delayed benefit. In the treatment for adolescents with depression study, which Bob Findling and the group here were a part of, we compared fluoxetine to cognitive behavior therapy, their combination, and pill placebo in adolescents with moderate to severe depression. And one of the ways that we were able to justify the use of placebo is that patients who were given placebo at 12 weeks, which was the longest that we felt it was ethical to hold a research subject in a placebo arm, were evaluated for whether they were non-responders or partial responders or full responders. Non or partial responders were offered the option of any of the active treatments in the trial, and the majority of them chose the combination arm from the study team in many cases. those who were responders to placebo, because most placebo responders actually don't do well over the long term, were offered, if they relapsed, the option of getting study treatment from one of the treatment teams. And you can see the outcome of this on this slide. This is from a paper published by Betsy Kennard last year this year in the American Journal of Psychiatry. This is the overall response rate, the number of subjects on the y-axis who achieved response for those that were in the active treatment condition at baseline. And you can see that they all did pretty well. We had about a 60% average response rate. And all we did was to shift the placebo responders' response 12 weeks down the road. They had exactly the same trajectory of response. It just happened 3 months later. So the argument was confirmed that there, for these subjects, was delayed benefit from trial participation, that they, in fact, were not harmed by participating in the study, other than the benefit was delayed. Now, let's go back and think about the placebo response from the point of view of what it looks like and what's driving it. If you look at an active treatment condition, you can have patients who respond, patients who don't respond. These are non-responders, are active drug failures. Of those who respond, some of them respond because the active drug ingredient in the active drug produced the expected benefit. Some responded because they got the placebo response, and some just spontaneously remitted, what we might think about as the simple passage of time effect. If you go back now and look at the placebo treatment, you've got non-responders to placebo. These are the expected placebo failures. And those that respond have the two conditions that are present in the active drug response not related to the active ingredient being tested, namely they're placebo responders or they are spontaneous remitters. So what we want to try to understand is what's going on with these guys here that contributes to the placebo response. And in essence, this is another way of asking the question, what is it that a placebo is controlling for? So when you have a comparator treatment, say a treatment as usual condition, or you plan to use a placebo, the question that needs to be answered is, what is this comparator treatment controlling for relative to your active treatment condition? And this, for a placebo, boils down to regression to the mean. If you take a look at a baseball hitter who hits 400 in a season, the chances are extremely unlikely that they're going to hit 400 in the next season. So what goes up comes down, and statistically, this is called regression to the mean. Spontaneous remission, you expect some patients will get well or improve simply because of the passage of time. Waxing and waning in the clinical picture. Many of the illnesses that we treat have unstable baselines, but people go up and they go down, not because they're remitting, recovering from their illness spontaneously, but because the baseline, the behavioral symptomatic baseline is unstable, and sometimes just by chance you're going to catch people on the upside of this instability profile rather than on the downside. Which is an experimental design and measurement question as well as a placebo question. And developmental changes. This is another passage of time issue, which is that people do grow out of these things, at least sometimes in psychiatry. So, for example, if you look at ADHD, the criteria we use, the prevalence of ADHD drops as you hit teenagers because they tend to be able to sit still better if you apply the criteria strictly. So there are developmental changes that happen that will look like improvement, but are in fact developmental. Now, for the most part, our trials are far too short for us to think about these developmental changes. But as we move toward trials which are asking questions that are on developmental trajectories, let's say, for example, we have a treatment which is preemptive, not simply symptomatic, that's disease-modifying, and the best end of the disease-modifying therapies is to prevent disease in the first place, then we will have very long clinical trials in order to be able to sort out these issues of disease modification and preemption, and there the developmental effect will come into play. Placebo also controls for time and attention and the impact of simply being in a protocol. If you look in a depression trial and you think about a psychotherapy condition like cognitive behavior therapy, we get people out of bed. That's called behavioral activation. We give them the expectation that something is happening which is good for them. That's a cognitive intervention. We ask them over and over and over and over again about all the different kinds of cognition changes that are characteristic of depression, for example, hopelessness, and it's embedded in the context that hopelessness is bad and it's a good thing if you aren't so hopeless anymore, which is essentially cognitive therapy, a watered-down version. So there are many, 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 many things in a clinical trial, including the human interactions between the research team and the subject, which are active ingredients of the placebo response, and in some sense they represent the kinds of things that are happening in the active treatment condition, but they're not formalized. So this is sometimes called the demand characteristics of a clinical trial, and involves time, attention, all the things that happen to patients across the different arms. And then expectancy effects are the ones that people typically talk about as important to the placebo response. And these are faith in treatment. People believe that they're going to benefit from being treated. So this is the kind of beliefs and wishes of the research subject. And they come from the fact that they're seeing somebody who is often one of the world's experts in the particular disease state that they're seeing. So there's authority, novelty, social approval. The process of informed consent can actually contribute to this faith in treatment by augmenting rather than confronting the therapeutic misconception. There's also a treatment provider bias that is part of this faith in treatment. Clinicians tend to expect that their patients are going to do better, partly because they're clinicians, and partly because they have the expectation in a placebo-controlled trial, you don't know actually whether you're getting active treatment or placebo, at least in theory. So you expect that your newer treatment is, because it's better, is actually going to benefit patients. And so there's this bias toward an expectancy-based improvement, and that contributes to the placebo response. And that's true even if the clinician is not doing the rating. So there's rater bias if you use, for example, independent evaluators, or you're using the subject, him or herself, as the rater, or the parent as the rater. There's a rater bias toward seeing benefits. And this is one of the reasons why people have argued that we should use centralized raters who do not know anything at all about the research subject. They don't know anything at all about that could help the rater peek through the blind about, say, for example, side effects. And even more so, you can use different raters for different subjects at different visits. So in fact, even the rater isn't the same, so they know nothing about the trajectory of that subject. They don't even know what clinical trial they're in. So if you wanted to get rid of all this stuff, centralized ratings is one way of doing it. And as I mentioned before, it's possible to operantly condition expectancy during the course of the clinical trial. That is, to reinforce the expectation of benefit. So we have this trajectory of improvement that if you know that, you will figure that your data point at week 6 is going to be predicated on your data point at week 4, and if the slope is downward, then you would tend to believe that your CGI score should be a point lower, more favorable at the next visit than compared to the previous visit. Adverse events can telegraph the blind, and the assessment protocol itself, as I talked about using the cognitive therapy example, can condition expectancy, cognitive expectancy effects. So Wired Magazine had a big article on the placebo response this year that was titled "Real or Fake, Both Can Cause Depression." So they have seen, as has everybody else, that there are these cohort effects in clinical trials in psychiatry where the placebo response rate has gotten larger over the years, making it much more difficult actually to get separation from the active treatment. And so we have many more failed trials or negative trials than we did previously. A negative trial is a trial against placebo in which there's no drug-placebo difference that emerges. A failed trial is an active comparator trial in which the active comparator, so you have the new treatment, the old treatment, the one that has been shown to separate from placebo previously, and then you have placebo. So in a failed trial, neither the new treatment nor the active comparator separate from placebo. And this is happening more and more often in our trials, even in trials in schizophrenia. So they wrote a big article on why this is happening, because it contributes to the $1.5 billion that it takes to bring a drug to market, and makes drug companies much more conservative about which drugs they are willing to promote into phase 3 trials. And so we learn less, and we have less available to our patients, and what we get is more and more kind of copycat drugs. So I won't spend any more time on this, but there are major implications for this effect with respect to kind of how we bring treatments to market. Now, this is the kind of conventional view of placebo, and it's all actually valid and credible and important. But there's been a fair bit of research over the last few years that is beginning to overturn this idea that placebo is just this inert thing, and it, in the brain anyway, doesn't do anything. And this is a study that was done in Parkinson's disease in 2002 that began this rethinking process, published in Lancet Neurology. And the trial was looking at apomorphine, which is a dopamine receptor agonist, in patients with Parkinson's disease. And they were using a positron emission tomography to look at receptor occupancy at the D2 receptor in the striatum, which is where the pathobiology of Parkinson's disease is located. And they had a hard time doing the trial because apomorphine makes people throw up. So they introduced a placebo group, and they modified the way they did the clinical trial. And what they found at the end of the day, when they looked at the PET data, was that if you got placebo, you had a surge in dopamine in the striatum, which was attenuated relative to the apomorphine condition, but which was meaningful, not trivial. And you can see that here, actually. This is at baseline. This is the extent to which the PET ligand bound to D2 receptors in the striatum, actually the putamen. And you can see this is lighter than this, which indicates that there's actually dopamine sitting on this receptor, so the PET ligand was outcompeted in the placebo condition. So that placebo was doing in the brain exactly what apomorphine was doing. It just wasn't doing it as effectively. It's an active treatment in the same circuitry, using the same molecular dissection methodology as the active treatment, not an inert pill.
Was there no treatment? Was there a no treatment group to compare to?
Not in this particular study. So I'll hold questions to the end. So this is a study done by Helen Mayberg at Emory, and what they did was to take a look at subjects that were getting in a randomized controlled trial context, either fluoxetine or placebo. These are adults with major depression. And what they did was they looked using functional magnetic resonance imaging. I'm sorry, they did this also with PET. And what they did was they looked to see if they could co-localize using regional changes in glucose metabolism. They could co-localize the effect of placebo and the effect of fluoxetine. And they found, in fact, that there was substantial co-localization. So if you look in the prefrontal cortex, the posterior cingulate, or in parietal association cortex, that there was substantial co-localization of the placebo effect relative to the active drug effect across the course of the clinical trial. So again, a demonstration using PET that placebo is doing the same thing as the active drug in specific brain regions. And then the most recent and really marvelous study that looks at this was done by John Zubieta at the University of Michigan. This is a complicated study looking at opiate and reward pathways in patients given a pain challenge. I won't go over the methodology of the trial, in part just to save time. But what they showed was that the placebo had exactly the same effect in the brain as an opiate agonist would have, and that this effect was not only mediated by the mu opioid system, it was mediated by the dopamine reward system in the nucleus accumbens, and that those patients who showed the placebo response had different behaviors on a gambling task, a reward-related task, than those patients who did not have the placebo response. So that if you were a placebo responder to opiate challenge in a pain context, you were more likely to show risky behavior in the gambling task, and that all this was mediated not only by the opiate systems, but also by the reward systems, tagging those two systems together in a circuit-specific model of the placebo response. So we have now an emerging body of knowledge, and there's actually quite a bit of research in this area that suggests that placebos are doing the same thing as the active drug in a relatively specific sort of way, so that in that slide I showed you at the beginning, where you're just looking at this sugar pill, is no longer really the proper way to think about placebo response. So to summarize that literature, it appears that if you interrogate synapses and circuits, that the placebo employs some sort of the same underlying brain mechanisms that are involved in the active treatment effect. The placebo activates this response. The nocebo effect may impair the response, which is what the John Zubieta study showed, and that this will vary based on the therapeutic area that you're looking at, which circuits are involved, which neurotransmitter systems are involved, which symptom domains are being looked at. And it's important to stay tuned to this research literature because it has significant implications for how we think about placebo and, for example, the issue of direct benefit. So let's just say that placebo response is a necessary evil, and now ask the question, how can we minimize it? And this begins with the definition of assay sensitivity. Assay sensitivity is a very specific word that means the property of a clinical trial that allows it to distinguish an active drug from a comparator. Without assay sensitivity, the trial's not valid for its intended purpose, and placebo is used largely to maximize assay sensitivity. When I talked with you about the cohort effect, what I was saying, in essence, was that assay sensitivity has gone down. So this is publication year and the effect size of the drug placebo from the drug placebo difference in depression trials, and you can see over the years that the assay sensitivity has been significantly degraded. Here's an example of this. This is two studies from Graham Emsley comparing fluoxetine to placebo in pediatric major depression. Here's the placebo response. Here's the drug effect. Downstream, this is the sertraline in pediatric major depression trial, and you can see that the drug effect was roughly the same and the placebo response rate was substantially enhanced. Now, this is not a property of the drug or the placebo. It's a property of the operations context in which these trials were done. That is, it was a sloppy clinical trial that was outsourced to a CRO and done in multiple sites globally. It just wasn't done as well as the Emsley studies. This is from a journal called Clinical Trial Magnifier, which, if you're interested in the clinical trials landscape across therapeutic areas, is a really useful thing to subscribe to. It's free. It's done in an institute in Hong Kong. And it shows you that for sites and for subjects, that industry trials have a lot more sites and they have a lot more subjects than non-industry funded trials. And yet these trials are likely to be more positive than these industry funded trials. And that is because NIH studies, even though they're sloppy in a good clinical practice sense relative to industry, have a whole lot of methodological niceties, one of which is to enroll appropriate subjects relative to industry funded trials. And And in this situation, this tendency to outsource everything has really degraded assay sensitivity. So if you think about this from an effect size point of view, you have the calculation being the mean of the active treatment minus the mean of the control treatment divided by the pooled standard deviation. So anything that makes this narrow or that widens the standard deviation will reduce assay sensitivity. So here's one way to get around it that I mentioned before, using video raters relative to clinicians and looking at the appropriateness of subjects for trials. So you would figure if you were screening patients that you should have a normal distribution of subjects entering the clinical trial. Some would get in, some would not, some would fall below a particular cutoff. In this case, we're looking at the HAM-D. This is the site ratings for subjects enrolled in a clinical trial, and everybody meets the lower bound HAM-D qualification score of 17. This is the central rating. And you can see the central rating gets a normal distribution. The investigators are enrolling a third of the subjects because they want to put people in. Sites get validated and paid for enrolling subjects, not for screening them out. And if you look at subjects overall, the effect size was 0.3. And if you just look in those subjects who actually should have been in the trial, it was 0.6. This is a failed trial. This is a successful trial. Now, that's the mean difference issue from what's called baseline inflation. If you now go to thinking about minimizing error, the denominator standard deviation term, if you have very low variability, you get nice separation between your two intervention groups. If you have high variability, you can see clearly that your standard deviation is going to go up, your effect size is going to go down, and it's going to have you're going to have much more difficulty with expected assay sensitivity. And so what we do is we try to select decent measures. We try to have measures that are stable. We spend a lot of time and money on inter-rater reliability and on reducing site variability, all to try to minimize error, contributing to a wider standard deviation. Now, if you think about this from the point of view of the value of these trials as credible experiments, this is the formula for the standard error of the mean. It's the standard deviation or variance structure for a particular, this is essentially the mean of the means of the clinical trials divided by the total N. So if the number of subjects gets very large, then the mean of the trial becomes a reasonable mean of for the population, basically because you're narrowing that standard deviation. So this is why we do meta-analyses. We take 20 or 30 trials and we summarize them in a single test statistic because any individual trial may be somewhere on this kind of broad range of what works. The other way to do it is to do much larger trials than we've been doing. And if you think about a Gallup poll, which typically has 1,000, 1,200 subjects, that's a pretty reasonable estimator of the general population mean for whatever they're looking at. The same thing would be true in clinical trials if we ran larger subjects rather than running a lot of small trials and then trying to meta-analyze them. Now you can see this from the point of view of assay sensitivity, how much power that you've got relative to sample size. At a sample size of 1,500, you can detect very, very small differences relative to a sample size of 25 in which you need a large effect. If your assay sensitivity is degraded or you've simply chosen for money reasons to run or feasibility reasons to run a small trial, it's going to be much tougher to find positivity at the end of the day. So I'd like to say that whatever our pilot data suggest relative to the effect size, we should probably take a standard deviation below that and use that value to power our studies so that we have fewer failed or negative studies. So when is a placebo necessary? Well, to control for expectancy, spontaneous remission, demand characteristics, the things we've talked about. It's necessary in the absence of a standard of care, at least some of the time. With any new agent, it's likely to be the best, if not the only way that we can make a causal assertion regarding the efficacy of that new intervention, so proof of concept or phase 3 trials. And it's necessary to define the adverse impacts of treatment, because you need a no treatment, not a no treatment, but you need a control condition that lets you separate adverse effects associated with the control condition from those associated with the adverse from the active treatment. So we look at the TADS trial again. This is the treatment response at 12 weeks, looking at ITT and observed cases. Here are combination and fluoxetine. It's medication that's carrying the variants. Here's CBT, and here's placebo. Without the placebo arm, it would have been actually quite difficult to make the assertion that these two treatments were the same, and that combination and that cognitive behavioral treatment was no more effective than a placebo condition. What would have been the conclusion from this trial is that CBT worked, it just didn't work as well. And now we know that in fact for these moderate to severely ill patients, if you do CBT over 12 weeks, it's not going to get you where you need to be relative to using meds. Now if you look at long-term effectiveness, you can see how this plays out. We had the placebo arm. For ethical reasons, they had to go out. Now at week 18, CBT catches up to fluoxetine. I would have loved to have made this the primary endpoint. It simply wasn't ethical. Combination treatment is at maximum medical benefit, gets there well ahead of CBT or fluoxetine alone, but the fact that we couldn't carry the placebo arm longer in this makes this assertion here that CBT works, it just takes longer, and this assertion that they all catch up to each other less credible. It's not as strong a recommendation as it otherwise might have been. No way to do this, but it illustrates the point. With respect to adverse events, this is the data from the FDA that was used to black box the SSRIs for suicidality in children. It's a slide from Tariq Ahmad, and you can see the odds ratios for most of these studies are to the right of 1. All placebo-controlled trials. Impossible to understand this other than in a placebo-controlled trial context. This is the TADS data, which was the best data available so far on this question, and you can see here that pre to post for suicidal ideation, that there's more ideation in the fluoxetine-treated kids than in the combo kids. This is one of the sort of major impacts of the TADS, that CBT may actually get rid of this SSRI-associated increase in suicidality. And you see this for suicidal events. Here's fluoxetine, a 2.5 increased relative risk relative to the combo condition, which is the same for CBT. So this is actually a strong recommendation because of the use of the placebo condition for an adverse event. The FDA position is that they don't require placebo arms. They like them, but they don't require them. But you do need adequate and controlled studies. So accept all kinds of study designs, including alternatives that can be used to maximize assay sensitivity. Now, people use placebo because it maximizes assay sensitivity. It reduces costs, so your sample size doesn't have to be as large as in an active comparator trial, and simply because the prevailing methodology, everybody believes you can't do it if you don't use placebo, which is in fact not true. Here's a quote from Bob Tabor from the FDA. He says they're ethical when delaying or omitting available treatment has no permanent adverse consequences for patients, as long as the patients are fully informed about their alternatives. You can minimize risk. We've talked a bit about this by unbalancing the randomization, by shortening, so fewer patients get placebo, by shortening the duration of exposure to placebo, by providing an extension that has no placebo, where patients are getting the active treatment if they responded, or by giving them the active treatment in the extension, by providing rescue options, by adding other treatments to background interventions. You can run, for example, a psychosocial treatment in the background or another drug treatment in the background, and by providing a very careful monitoring plan, including using a data safety and monitoring board. Now, quickly about alternatives to placebo. This depends entirely on study stage. So if you look at the cycle of quality, where you move from discovery science through early translational steps, basically moving to first-in-man studies, clinical trials, phase 1 to 4, clinical practice guidelines, performing measures. This is where the learning healthcare system happens, and outcomes, and then you feed that back. This cycle of quality, you would see placebo in the early phases of clinical research. Now, if you look at those early phases, whether you're going to use a placebo or not depends on whether you're running an explanatory or a pragmatic trial. Typically, in our studies, like the TADS, we've blended efficacy and effectiveness elements, these hybrid designs. But the field is moving toward bifurcating explanatory and pragmatic studies. I know this because I'm currently chairing a council work group on interventions research, which is likely to make this recommendation. So pragmatic trials, or what are sometimes called practical clinical trials or large simple trials, help users choose between options of care. These are trials for decision makers. They're not designed for assay sensitivity in the sense of wanting to see if an active ingredient is different than not having that active ingredient. Explanatory trials, on the other hand, test causal research hypotheses. That is, that a given intervention has a particular benefit associated with the active ingredient of the treatment. And often, this is pegged to a mechanism question so that you understand the mechanism by which the active treatment is in fact working. In our studies so far, the extent that we include explanatory effects in pragmatic trials, it impairs generalization, and these pragmatic trials are too small to identify tailoring variables. They're also not suited for looking at mechanisms. So I think the field is going to move toward this bifurcation, and this is early phase research, phase 1, 2, and 3, where we are going to see placebo-controlled trials. Now, you can reduce exposure by modifying randomization, exposure to placebo. One way is to do a play-the-winner design, and that's you simply bias the coin toss based on response. So if you enroll your first 20 subjects, you see that the active treatment is beating the placebo treatment, you bias the coin toss so that patients are more likely to receive, or subjects, the active treatment. You can do adaptive randomization to determine the optimal sample size for assay sensitivity. So you can do interim looks at the data, protecting the overall alpha, and either adjust the sample size up or downward so that at the end of the day you have a trial which gives you a definitive answer relative to the experimental question being asked. You can use patient preference designs in which patients have a say in what kind of treatment they get, and one example of this is an equipoise stratified randomization in which you allow patients to allow or disallow particular arms, including disallowing the placebo alarm. These are used in pragmatic trials, not explanatory trials. You can do placebo discontinuation trials. Bob Tabor loves this. We haven't done anywhere near enough of these. These ask a different question. They're asking in patients who respond to a treatment, is efficacy preserved if you randomize patients to placebo as compared to the continuing on the active treatment? But they're more palatable than initial placebo randomization. And they may work if you've got circumstances where this happens organically. Patients take drug holidays, for example. It helps a lot with recruitment and the ethical questions. You can use an endpoint like need for rescue or all-cause discontinuation in these studies, which is, in some sense, a more clinically useful endpoint. They require a larger N, which is a downside, and it's sometimes a little tougher to blind clinicians and investigators. This is an example of a placebo-controlled discontinuation trial in kids with depression, again from Emsley's group, and here were responders, 100% of course, responders randomized to fluoxetine, and their relapse rate was lower than those randomized to placebo. And this would be an adequate efficacy demonstration for one of the two FDA registration trial requirements at phase 3. It's just not used very much. Add-on designs also work. You can have randomization to new treatments versus continuing on a previous treatment. This is the cancer epilepsy model, and there are some ways that this can be helpful and other ways in which it's not so useful. This is an example. First time you all will have seen this. It was presented twice now in a study that we did looking at patients with OCD who were partial responders to a serotonin reuptake inhibitor. We held our medication stable in the medication management condition. We added a watered-down version of CBT from a child psychiatrist who is doing the med management. This is the red line, and we did the standard dual doctor model, and what you see is that if you want to do this, you're going to need expert, more intense CBT in order to benefit. So this is an example of an add-on design that gives you clear assay sensitivity, not with a placebo condition. You can do treatment as usual and not use a placebo at all. This would be, for example, randomizing, as we did in the MTA subjects, to get whatever's available in the community. The downside is that you may have access barriers, or there may be great heterogeneity of practice from these community-based treatments. That makes it actually much difficult to find an effect because the standard of deviations go up, and it also is a credibility issue. If there was a lot of access barrier at site X, and a quality barrier at site Y, you would have a hard time interpreting the result. And at site X, with the access barrier, is it really a fair test if people can't get any sort of treatment? On the other hand, it's the ultimate pragmatic question, because what you want to do from an effectiveness or pragmatic point of view is to show that your new treatment is actually going to impact health outcomes in a way which is relevant to healthcare decision makers. You can also use active comparator designs. These can be dose-controlled, non-inferiority designs, or you can compare the innovative treatment to a standard of care or an active comparator treatment, but at a small test of differences. This is the Phil Lavori argument. I actually like this argument a lot, particularly for pragmatic trials, and that is if the new drug can't beat the old drug on benefits, acceptability or tolerability at a 10% test of difference, who the hell needs it anyway? So the idea here is not to do non-inferiority trials, but to go ahead and ask the clinically relevant question, is it good enough relative to something that's already available? So to conclude, and I'm sorry I ran over a little bit longer, I think it should be clear that there's an affirmative ethical mandate to conduct medication trials across the age range in psychopharmacology. To be ethical, these trials have to reflect sound scientific principles, methods, and rigor, including informed consent, and they must be fully justified, the placebo arm in a trial, including the presumption there's no substantive risk that the use of placebo will harm participants. These trials can't be used, placebo can't be used in a clinical trial if there's a safer and equally useful research technology available to answer the scientific question. But it should be employed if it's needed to answer the scientific question. That is, it can be answered in no other way and when the risk is acceptable. The notion that placebo is inert isn't any longer credible, and scientific basis for the neurobiological basis for the placebo response needs to be an active area of investigation, because it's part of what we do in an important way, and it has serious major ethical implications with respect to how we think about placebo. So these are my sort of acknowledgments. This is team science at its best. There are hundreds and hundreds and hundreds of people that have been involved in all of these projects. These are the funding sources. And from my point of view, the merit, if there is any, to doing this kind of work belongs to the children and families who participate. The whole edifice of medicine is based on a very small number of researchers and a slightly larger number of subjects. All of us benefit from their participation, and we actually need to treat them respectively and honor their contributions. Thank you.